Good morning, students. Our today's topic is lysosomes. These are membrane bound vesicular structures formed by the process of packaging in the Golgi apparatus. So, lysosomes, they are sac like structures formed by the Golgi bodies. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolysis like lipases, proteases, carbohydrates. So, in the lysosomes, they are very rich in enzymes, or in them, lipases, where the word lipases means they are lipid breaking or digesting. Proteases means they can digest proteins. Carbohydrates means they can digest or break carbohydrates. Where ases word is there, ASES, that means breaking. So lipases means they break lipids or fats. Proteases means they break proteins, and carbohydrates ases means they break carbohydrates at optimum acidic pH value. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. For nucleic acids, nucleases enzyme is present. Next is vacuoles. The vacuole is a membrane-bound space found in the cytoplasm. So, vacuoles also are covered by the membrane. It contains water sap, excretory product, and other materials not useful for the cell. Mainly the vacuoles contains the wasteful materials. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuole is large and it contains tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. So, large vacuoles are present in plant cells. In plants, the tonoplast, that is the membrane of the vacuole, facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is slightly higher in the vacuole than in the cytoplasm. Active transport means here ATP is used. Energy in the form of ATP is used in this. So movement is against concentration gradient from lower concentration towards higher concentration. In vacuole, the concentration is higher as compared to the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. Osmoregulation means maintaining water salt balance in the body. Excretion for removing out toxic material from the body. Can you tell me the difference between excretion and egestion? Yes, excretion is removing the toxic waste from the body. Egestion is removal of the fecal matter from the body. In many cells, as in protists, food vacuoles are formed by engulfing food particles. In protists, the food vacuoles are formed by taking the food particles from outside. Next is mitochondria. Mitochondria, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending upon the physiological activity of the cells. The cells which are more active, they have more number of mitochondria as compared to the which have less activity. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of 0.2 to 1 μm and length 0.1 to 4.1 μm. So their shape is sausage like. Each mitochondria is double membrane bound structure. Each mitochondria is surrounded by two layers, double membrane, with the outer membrane and the inner membrane, dividing its lumen means cavity, distinctly into two aqueous compartments. So outer membrane and inner membrane. The inner membrane divides the inner open cavity, inner side, into two parts, the outer compartment and the inner compartment. Outer means between the inner membrane and outer membrane, and between the inner membrane. The inner compartment is filled with a dense homogeneous substance called matrix. Inside the inner membrane, matrix is present or cytoplasm is present. The outer membrane forms the continuous limiting boundary of the organelle. Outer membrane is without any divisions or without any folds. It is smooth, continuous. The inner membrane forms a number of infoldings called cristae. The cristae increases the surface area. So what is the function of cristae? It increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possess circular DNA. So in the mitochondria, in this matrix, some circular DNA also is present. A few RNA molecules, ribosomes, they have 70 S type of ribosomes, and the components required for synthesis of protein. So mitochondria have their own DNA also and can manufacture their own proteins. The mitochondria divide by fission process. Next is plastids. Plastids are found in all plant cells and in euglenoids. Plastids mainly give color to the plant, so mainly they are present in all the plant cells. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific color to the plants. Based on the type of pigments, plastids can be classified into chloroplasts when they give green color, chromoplast when they give colored other colors, and leucoplast, they are mainly colorless. The chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy, which is essential for the process of photosynthesis. So chloroplasts have chlorophyll which trap the light energy. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophyll and others are present. In chromoplast, because they give different colors, so different pigments are present like carotenes, xanthophylls, etc. This gives the part of the plant yellow, orange or red color. The leucoplasts are the colorless plastids of varied shape and sizes which store nutrients. So in leucoplasts, their main function is storage. A myeloplast store carbohydrates or starch. Example is potato. Ileoplast store oils and fats, whereas the aleuroplasts store proteins. Now just see the diagram of the chloroplast shown here. Outer membrane, then inner membrane. In between, grana is present. Single part is known as thylakoid. A group of thylakoid combined together is known as granum. And in the matrix is in between, matrix also is present, that is known as stroma. The two granum are connected with each other by stroma lamellae. Majority of the chloroplast of the green plants are found in mesophyll cells of the leaves. Mesophyll cells means the middle cells, green cells. Upper epidermis, lower epidermis in the leaf, and then middle part contains mesophyll cells. So here chloroplasts are present. They are lens-shaped, oval, spherical, discoid or even ribbon-like structure in different organisms. Their number varies from 1 per cell in the Clydomonas, a green alga, to 20 to 40 per cell in the mesophylls, that is in the leaf. Like mitochondria, the chloroplasts are also double membrane bound. They also have double membrane. Out of the two, inner chloroplast membrane is relatively less permeable. Outer is more permeable and inner one is less permeable. The space limited by the inner membrane of the chloroplast is called stroma. The inner part of the inner area is known as stroma. A large number of organized flattened membrane sacs called thylakoids are present in the stroma. In the stroma, small disc-like structures, flattened membrane-like structure known as thylakoid. Thylakoids are arranged in stacks one above the other like the piles of coins and called grana. All the thylakoids together known as they form grana. Thylakoids are arranged in stacks like the piles of coins called grana or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoid of the different grana. The different grana are connected with each other by stroma lamellae. The membrane of the thylakoid encloses a space called lumen. In each thylakoid, the internal space is known as lumen. In the thylakoid, internal space which is present, that is known as lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. So in the stroma, again enzymes are present which help in the synthesis of carbohydrates and proteins. They also contain small double-stranded circular DNA molecule and ribosomes. Like in mitochondria, here also circular DNA and ribosomes are present. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, that is 70S, than the cytoplasmic ribosome. In eukaryotic cells, 80S ribosomes are present. S is means Sydberg constant. 80S ribosomes are present in eukaryotic cells, and in case of chloroplast, in case of plastids and mitochondria, 70S ribosomes are present. I hope this is clear. Next time we will continue with the next topic. Thank you.